In this activity, we're focusing on leaf abscission, the controlled process by which plants shed their leaves. You'll be looking at a prepared slide showing the abscission zone and learning how plants separate a leaf while still protecting the stem. Leaf abscission isn't random damage or tearing. It's a controlled, programmed process that happens in a specific region called the abscission zone. The plant actively prepares this area so the leaf can detach safely. It's important to remember that leaf abscission doesn't just happen because it gets cold or dry. It's actually regulated by plant hormones. Two of the main hormones involved are auxin and ethylene. When a leaf is healthy and active, auxin levels coming from the leaf help prevent abscission. As the leaf ages or conditions change, auxin levels drop and the tissue becomes more sensitive to ethylene. Ethylene then promotes the formation of the separation layer in the abscission zone, where the cell walls and middle lamellae begin to break down. So abscission is really about a change in hormone balance that tells the plant it's time to let this leaf go, while still making sure a protective cork layer forms to seal off the stem afterward. In the abscission zone, two important things happen. On the stem side, the plant forms a corky protective layer. On the leaf side, a separation layer develops where the cell walls and middle lamellae start to break down. As those connections weaken, the leaf eventually detaches from the stem. The cork layer is critical because once the leaf falls off, the plant is left with an open surface. That cork layer seals the stem, helping prevent water loss and keeping pathogens out. So abscission isn't just about getting rid of a leaf, it's also about protecting what's left behind. As you think about this question, focus on what problem the plant is solving. What would happen if a leaf just tore off randomly? Think about protection, water loss, and disease entry. Write your prediction in your lab notebook before you start the observations. You're observing a longitudinal section through the abscission zone of poplar under low power. Start by finding the petiole, which is the stalk of the leaf. This gives you your orientation. Next, look where the petiole meets the stem. That region is where you'll find the abscission zone. Running through this area, you should see the vascular bundle, which connects the leaf to the stem's transport tissues. Near the node, look for the axillary bud, which sits just above where the leaf was attached. Covering the bud, you may see bud scales which protect it during dormancy. As you look at the image, you'll make a sketch in your lab notebook and label these structures. Use their positions as clues. Notice where the petiole connects to the stem, where the vascular tissue runs, and where the bud is located relative to the abscission zone. For the first question, think back to activity 1. Where did leaf scars come from? Now connect that idea to what you're seeing here in the abscission zone. Ask yourself, how does one lead to the other? For the second question, focus on protection. What risks does a plant face when tissue is damaged or removed? Then compare that to what happens when an insect chews into a stem or leaf. In both cases, the plant needs to seal off the exposed area, but the cause of the damage is different. Leaf abscission is a great example of how plants manage damage and seasonal change in a controlled, protective way. What you see internally in the abscission zone explains what you later see externally as leaf scars on woody twigs. Once again, this is all about connecting structure and function in plant anatomy.